In 1869, Friedrich Miescher isolated an acidic material from the nucleus of a cell and called it nuclein, now known as nucleic acids. Also around the same time, Gregor Mendel proposed the principles of inheritance. In fact, Gregor Mendel, Thomas Hunt Morgan, Walter Sutton, and numerous other scientists had narrowed their search for genetic material to chromosomes. However, as chromosomes of eukaryotes contain several proteins along with DNA or deoxyribonucleic acid, scientists couldn't exactly confirm whether the genetic material was DNA or proteins or both. By around 1926, the search for the mechanism of inheritance of genetic material had reached the molecular level, yet the genetic material had still not been discovered. However, in 1928, Frederick Griffith, a British scientist, conducted an experiment that accidentally demonstrated the transformation of genetic material in bacteria. In fact, Frederick Griffith was in the process of developing a vaccine against the bacteria Streptococcus pneumoniae that caused pneumonia by inoculating the dead microbes along similar lines of Louis Pasteur's experiments on vaccination. Frederick grew the bacteria in a culture plate and found that they were of two forms, the smooth and the rough. This was because the smooth, shiny colonies or the S strain of the bacteria possessed a mucus or a polysaccharide coat, while the rough colonies or the R strain had no such coat. Further, when the mice were injected with the S strain, they died of pneumonia infection, while those injected with the R strain did not develop pneumonia. This proved that the S strain of bacteria was virulent or lethal, while the R strain was harmless. Moreover, Frederick also found that heating killed the bacteria. So when he injected heat-killed S-strain bacteria into the mice, they did not die. On the other hand, when he injected a mixture of R-strain bacteria and heat-killed S-strain bacteria, the mice died. Surprisingly, he even recovered the living S strain of bacteria from the dead mice. So on the basis of this experiment, Frederick concluded that the heat-killed S strain bacteria had somehow transformed the R strain bacteria. He also concluded that the reason why the R strain bacteria produced a smooth polysaccharide coat and became lethal or virulent was because of the transfer of some genetic material from the heat-killed S strain bacteria. However, there was still no means to ascertain or identify the genetic material that was transferred and responsible for the transformation process. Later, after 16 years, in 1944, a team of 3 scientists, Oswald Avery, Colin MacLeod, and Macklin McCarty once again began research on the experiment that was once conducted by Frederick. At that time, they tried to identify the biochemical nature of the transforming principle in Frederick's experiment. For this, the scientists first purified biochemicals such as proteins, DNA, and RNA or ribonucleic acid from the heat-killed S-strain bacteria. They did so to identify the biochemical responsible for transforming the live R strain into S-strain bacterial cells. Through their experiments, they discovered that it was the DNA from the S-strain bacteria that had transformed the R-strain bacteria. They also found that digestion of the DNA of heat-killed S-strain with DNase inhibited transformation. Further, the scientists also discovered that addition of proteases or protein-digesting enzymes and RNases or RNA-digesting enzymes did not affect the transformation. This led to the conclusion that proteins or RNA were not transforming substances and implied that DNA was the cause for the transformation. Thus, through their experiments, Oswald Avery, Colin MacLeod, and Macklin McCarty concluded that DNA is the hereditary material in most living organisms. Nevertheless, many biologists were still unconvinced that DNA is the hereditary material. And so the search for genetic material continued for several years and was concluded with the discovery of DNA. In the past, it was believed that proteins were the genetic material. However, in time, through the various experiments using bacteria and viruses, the truth about genetic material came to light. British bacteriologist Frederick Griffith was the first to conduct experiments with bacteria Streptococcus pneumoniae that was responsible for causing the disease pneumonia. He worked with two types of bacteria strains, the pathogenic S strain and the non-pathogenic R strain. The S strain has a polysaccharide coat that makes it smooth, whereas the R strain lacks it, making it rough. During his experiments, Griffith realized that when heat-killed S-strain cells were mixed with R-strain living bacteria, and this mixture was injected into healthy mice, they died of pneumonia. Thereafter, when the bacteria were isolated from dead mice, they were found to be of the living S-strain type. This proved that the R-strain bacteria had transformed to the living S-strain. Yet for a long time, due to lack of conclusive proof, proteins were believed to have caused this transformation. However, in 1944, Oswald Avery, Colin MacLeod, and Macklin McCarty worked together to determine the biochemical that had caused the transformation in Griffith's experiment. They repeated Griffith's experiments and purified all three biochemicals such as proteins, DNA and RNA from heat-killed S-strain cells to identify which one had caused the transformation. They discovered that neither the protein-digesting enzymes, that is, proteases, nor the RNA-digesting enzymes, that is, RNases, affected this transformation. Whereas DNA digestive enzyme DNase did inhibit the transformation. Therefore, it was clear that the transforming substance was not protein or RNA, it was DNA. 8 years later, in 1952, the definitive evidence that DNA was a genetic material was provided by Alfred Hershey and Martha Chase. They conducted their experiments with viruses that attack bacteria, better known as bacteriophages. Bacteriophages attach to bacteria and implant their viral genetic material inside the bacterial cell. The bacteria treat this genetic material as their own and produce more of such virus particles. Since Hershey and Chase wanted to find out whether it was protein or DNA from the virus that entered the bacteria, they grew these viruses on two different mediums. One with radioactive phosphorus and the other with radioactive sulfur. As DNA contains phosphorus, while protein doesn't, the viruses that were grown in the presence of radioactive phosphorus had radioactive DNA and not radioactive proteins. On a similar note, as DNA doesn't have sulfur like proteins, the viruses grown on radioactive sulfur had radioactive proteins and not radioactive DNA. Thereafter, both these radioactive phages were allowed to infect E. coli bacteria. Later, after the infection, when viral coats were removed from the bacteria, it was revealed that the bacteria that were infected with viruses containing radioactive proteins were not radioactive. On the other hand, the bacteria that were infected by viruses with radioactive DNA were radioactive. It was clear then that proteins did not enter the bacteria from the viruses. Rather, it was the DNA that passed on from the virus to the bacteria. Therefore, despite experiments by Griffith and Avery, MacLeod and McCarty, it was the experiment by Hershey and Chase that proved the fact that genetic material is DNA.